The neutral theory of molecular evolution, which was proposed in the 60s, states that most DNA mutations are neutral and spread through populations by chance rather than by natural selection. This theory is supported by the observations, which are non-fictional segments of DNA that evolve the fastest due to their accumulations of neutral mutations. This aligns with the law of increasing entropy, suggesting that random DNA mutations significantly influence the rate of evolution across different species. While this theory adequately explains the molecular evolution rates of many organisms based on random motion, understanding the evolution of neurons might require considering additional factors such as electrical magnetic forces and quantum entanglement. Neurons might have adapted the environments rich in electromagnetic rhythms and utilize quantum entangled states in specific noise-free conditions. The evolution of brain-expressed genes is notably slower than that of genes expressed in other tissues, a phenomenon likely due to high level of functional constraints. One such constraint might be the integration of information by neurons' assemblies, enhancing environmental adaptability. This particular study explores the physiological mechanisms of information integration in neurons through three types of synchronization: chemical, electromagnetic, and quantum. Chemical synchronization involves the diffuse release of neurotransmitters like dopamine, for example, causing transmission delays of several milliseconds within neurons. Electromagnetic synchronization encompasses action potential, the electrical gap junctions, for example. These electrical gap junctions enable rapid synchronization within the cortical of GABA network, while apoptic coupling allows structures like axons bundled to synchronize through extracellular electromagnetic fields, surpassing the speed of the chemical processes. Quantum synchronization is hypothesized to involve ion coherence during ion channel passage and the entanglement of photons within the myelin shield. Unlike the finite time synchronization seen in the chemical and electromagnetic processes, the quantum entanglement provides instantaneous, non-local synchronization and coherence states within the brain tissues. Neurons might have evolved from slower chemical infusion to rapid temporal synchronization with ion passage through the gap junctions within the cortical of these networks of neurons. This particular study is a mini review that compiles different literature on these three synchronization types, offering new insights into the physiological mechanisms that address the binding problem in neuron assembles. To understand molecular evolution in relation to higher order functions like information integration in cortical neural circuits, it's essential to address the binding problem. This problem involves the physiological mechanisms by which the brain integrates information processed in different locations, such as color, shape, and motion, into single perceptual experience. Two major hypotheses addressing the binding problem are the synchrony hypothesis and the firing rate enhancement hypothesis, where these two different hypotheses are discussed extensively in many different research papers. To comprehensively understand the molecular evolution, though, related to information integration in neurons, it might be then necessary to consider not only random motion, but also regular motion induced by electromagnetic forces and state changes due to quantum entanglement. Solving for the information binding within the neurons requires us to understand how these neurons assemble and how they have evolved their ability to synchronize information through the three synchronization methodologies that we just talked about, through the chemical assembly, the electromagnetic synchronization and assembly, as well as the quantum synchronization and assembly. Within the electromagnetic synchronization, the speed comes into play because it happens much faster than in comparison to the chemical. And when it comes to the quantum synchronization, it also explores the way in which it could solve potentially the information binding within the neurons and how they assemble such that we can understand the world around us and that we can process and form and extract information within and without us. Let's first look at the slow chemical synchronization, aka the neurotransmitters within the human brain. The emergence of neurons represent a pivotal evolutionary development, but the precise mechanisms of their initial evolution remains uncertain. Approximately 600 million years ago, the ancestors of neurons likely began communicating through the secretion of neuropeptides containing vesicles, despite lacking the ability to generate electrical activities. These primitive neuropeptides secretions would have induced contraction responses in nearby cells, allowing rapid reactions to external stimuli. Consequently, early neurons might have primarily utilized chemical synchronization through volume transmission, disseminating information slowly but simultaneously to multiple. This type of chemical synchronization through volume transmission persists in the mammalian brain. Volume transmission involves the diffusion of neurotransmitters such as dopamine or serotonin, and it comes from the brainstem in most cases, and it controls activities such as sleep, wake rhythms, emotions, and so forth. Over evolutionary time, the slow chemical synchronizations of peptide-based neurons might have evolved into faster electromagnetic synchronization through synaptic transmission involving action potentials, where the neurons become hyperactive and then they stop. So it's like zeros and ones essentially. This evolution improved the precision of information transmission, leading to synaptic wiring characterized by fast and specific one-on-one communication within the neurons. Modern neural networks incorporate both slow chemical, such as neurotransmitter, and fast electromagnetic synchronization. Now, let's look at the fast electromagnetic synchronization within the brain. This high precision in action potential in the neurons enhance memory capabilities through spike timing dependent synaptic plasticity. Even a 1 millisecond delay can shift the phase of gamma oscillations, aka brainwaves, significantly affecting the synaptic plasticity of the brain. Thus, the temporal control of information through action potential propagation along axons is a crucial mechanism for synaptic plasticity. In this case, the neurons dramatically increase the conduction speed within themselves, optimizing information transmission efficiency in white matter composed of these axons within the brain, influencing the control and the synchronizations of axonal signal transmission and affecting alpha brainwaves peak frequencies, enhancing chemical and electromagnetic synchronization within the brain is crucial for brain evolution. In terms of electromagnetic synchronization within the neural network of the brain, various physiological mechanisms such as synaptic transmission through action potential, GAMP junctions in inhibitory GABAergic neurons, and other type of phenomena might play a role in strengthening the synchronization of information within the brain. To integrate information across complex brain system into a unified whole, the way we know it and the way in which we perceive the world, is an essential to consider not only conventional chemical and electromagnetic synchronization, but also the quantum synchronization, which facilitates the instantaneous information transfer regardless of the distance within the brain. Now, let's look at the instantaneous quantum synchronization research within the brain information processes. Quantum coherence was previously thought to be incompatible with biological systems due to environmental noises or environmental impact. But its potential role in various biological processes is now gradually being recognized. For example, electron energy transport in photosynthesis or within the birds who migrate, which is a part of the discussion within the, for example, field of quantum biology. And one hypothesis suggests that, for example, different molecules composed of calcium and phosphate within the neurons might contribute to the factor that there is a information exchange at the quantum level that might be happening also within the neurons, which could also potentially help with the transmission and storage of information within ions selectively in ion channels also play a crucial role in the evolution of neurons by enabling precise control over the flow of the electrical signals within the neurons. This selectively allows specific ions, such as sodium or potassium, for example, to freely pass through the small pores in the cell membrane. The physiological mechanisms underlying this selectivity are understood through physical mechanisms such as the size relationship between the channel pores and the ions, as well as hydration by water molecules. Potassium ion channels, for example, are regulated to allow only potassium ions to flow across the membranes of the neurons, which preventing other ions such as sodium from going in and out. Despite the selective filter region within the channel allowing only potassium ions to pass through, the selectivity is remarkably high, allowing up to 100 million potassium ions per second while only one sodium ion can pass for every 10,000 potassium ions. So the physiological mechanism involving potassium ions surrounding the water molecules within the neurons is also extremely important to think about during this information processing ways in which neurons form information, transfer information, and also interpret information. There appears to be also a consistency between the electromagnetic versus the quantum synchronization within the brain. Theta gamma phase amplitude coupling in EEG is associated with working memory, while beta gamma phase amplitude coupling is related to motor functions. This suggests that in phase amplitude coupling, the amplitude of high frequency bands, let's say gamma, contains more information than the phase of low frequency bands, let's say beta brainwaves. And action potential along axons alone cannot fully then explain the differences between high frequency and low frequency brain waves in solving the information binding problems in the neurons, as action potential along axons can also contain low frequency bands such as alpha or theta. So in trying to understand how the human brain evolved to what it is today, the neurons might have evolved to enhance their adaptability to the environment by integrating information through chemical, electromagnetic, as well as quantum synchronization. Chemical synchronization, that is where the neurotransmitters have a role in exchanging and transferring information, is the slowest way for the human brain to process and integrate and move information. However, through evolution, the human brain needed a faster way to accumulate and transfer information. And that is when it apparently evolved into also using the electromagnetic synchronization of information transfer and processing within the brain. And that is when action potential comes in, the electrical activities of the brain comes in, and that's where the brain waves frequencies such as alpha, beta, gamma, and so forth come in to help with information transfer, information translation, information processing, and lastly, in addition to the chemical and the electromagnetic synchronization of information within the brain, most recently the quantum synchronization has also been proposed and studied by many research scientists. In this case, it involves instantaneous information transfer and translation, and it happens within the instantaneous way in which the neurons would then communicate with one another. This includes ion coherence through selective ion channels and entangled photons, light molecules, within the axons. Quantum entanglement offers the unique characteristics of instantaneous transmission of information regardless of the distance in which the information needs to be transferred. Understanding the evolutionary origins of neurons synchronization then might help us understand all these neural circuits that are responsible for integrating information into a unified way in which we understand ourselves, understand the world, and bottom line, create and process and transfer information at the neural. I'll share the link to this article below so you can read it yourself because it's very long, but it's extremely exciting because this is one of those papers where they have tried at least to accumulate and look at all the different types of research paper that exists about understanding the brain as well as its evolution. Hence the reason they talk about these three different ways in which the information is being synchronized within the brain. And so what they're trying to say here is that it's great that we're trying to understand neurons and human brain from conducting all these different studies, but it's also important to look at it as a whole, because the way in which information is even presented at that level of neurons is still an enigma to us. And as you might know, it's been one of the questions I've had since I don't even remember how long, and we're still in the infancy stage of trying to understanding it. But these are a few scientists from University of Toyama in Japan, who's done the accumulation of these different research papers. And obviously they cite these research papers if you are interested. But definitely these conversations, at least at a scientific level, started back in the 60s, and we're still continuing with that. And that is the theory that I mentioned in the beginning of this video called neutral theory of molecular evolution. That's all I have for today. Thanks. And yeah, feel free to like, share to increase the engagement for this video. Take care. Bye.